Hello, welcome to Converging Dialogues. This is Xavier Bonilla. On this episode, I am very honored and very pleased to bring the conversation I had with Venki Ramakrishnan. Venki is the group leader at the Medical Research Council Laboratory of Molecular Biology at the Cambridge Biomedical Campus, United Kingdom. He's also a fellow of Trinity College, Cambridge, and was president of the Royal Society from 2015 to 2020. He has a background in physics. He has a Bachelor's of Science in physics. He also has a PhD in physics, but was then made the switch to biology. And he has been doing biology for some time. What is most notable, he's had many awards and many accolades. He is most notably known for winning the Nobel Prize in chemistry in 2009, along with Thomas Stites and Ada Yonath. And he has also received India's second highest civilian honor. He's also been knighted in 2012 for his services to molecular biology. He's in 2020, he was elected to American Philosophical Society and board member of the British Library. He's also a member of the Order of Merit as recently as 2022. So he is, not only is he a Nobel Prize winner, but he's also a knight, and he has done so many things. And the reason for all of these accolades is that he was highly influential and instructive in the structure and detailing the structure of the ribosome for the cell, which is absolutely incredible. And it was quite a joy to talk with him. He is, exceptionally brilliant, but one of the most humble and down-to-earth people I've probably ever talked to. And he wrote a book a couple of years ago, and the paperback was just released, entitled Gene Machine: The Race to Decipher the Secrets of the Ribosome. And so it's a quasi-autobiographical and then quasi kind of scientific, which is really wonderful. And in it, he talks about, in the conversation, we talk about how he came from India, and he was studying physics there and came to the United States and studied physics. And then, in the middle of the PhD program, he said, physics isn't for me. And he wanted to do biology, and he made that switch. We talk about the layout of the ribosome. We talk about DNA, RNA, mRNA, and proteins. We talk about when he first got started in studying the ribosome in the late 70s and early 80s, what was known then, and then what was his kind of contribution and how we've learned so much up to the present and how we were able to get the structure of the ribosome. We spent some time talking about X-ray crystallography and his trip to the LMB for the crystallography. We talk about his experience of seeing atomic subunits of ribosomes for the first time and what that was like as being one of the first people to ever see that and to really observe that. He lays out in the book and in the conversation the experience of being nominated for the Nobel Prize, what that's like, how they inform you, attending it, what it's, just the whole thing. And it was like a kind of peeling back the curtain a bit. And how it is to win a Nobel Prize, but what the experience is like, which was really fascinating. And really, he talks about it in the conversation, but how your life kind of changes afterwards, which is really interesting to think about. And then we talk about the future of ribosome research and where that's going, and more importantly, why it's essential for understanding many things in medical science, but also for potential cures for things and being a contribution there. So it's all very important. As always, you can find this conversation and all other conversations at convergingdialogues.substack.com, also on YouTube. So please follow, like, share, and subscribe and contribute as much as you can. And telling your friends always helps. And make sure you go out and get Venki's book. It's quite wonderful. And now I bring you Venki Ramakrishnan. I am here with Venki Ramakrishnan. Thanks so much for coming on the podcast. I'm very much looking forward to speaking with you.
Thank you. It's a pleasure to be here.
Yes, absolutely. So you are known for many things. One of them is the book that you have written, which is now in paperback, called Gene Machine: The Race to Decipher the Secrets of the Ribosome. You're also known for winning the Nobel Prize in chemistry. You're known for understanding the structure and laying it out of the ribosome, which is amazing. So you're known for many things, which we'll get into. I guess the first question I want to ask is for people that are unfamiliar with you and your work and stuff. What's kind of the snapshot of who you are professionally, academically, and what you currently spend your time doing?
Yeah, so I'm what you loosely call a molecular biologist. I like to understand the molecules that are at the heart of life. And in fact, from 1978, when I was still in my 20s, I settled in on a very complicated molecule that's at the heart of what makes us alive. And that's this large complex called the ribosome, which is the machine that translates the information in our genes to make the proteins that are coded by those genes. So, if we say everybody thinks they know what DNA is, they think, oh, yeah, it's about our genes and so on. But then you ask them, well, what is a gene? And they'll say, oh, well, it's those characteristics we inherit or pass on, and you can have good genes or bad genes. That's about as far as most people get. But then, so what are these things? So it turns out genes are basically coded information which reside on our DNA. And that has to somehow be used to make all the different proteins that make up life. So the reason you're able to see me or see the screen is because there's a protein in your eye that senses light. The reason that you can breathe oxygen and deliver it to your muscle is there's a molecule called hemoglobin, a protein called hemoglobin, which takes the oxygen in your lungs, goes through your blood, and delivers it to the muscles. So, and antibodies, when you get an infection, you generate antibodies. Those are all proteins. The fact that you can hear is due to other kinds of proteins, or touch, or smell. They're all due to proteins. Memory is formed by proteins. So proteins do a huge variety of things, and every one of them is made using information encoded in our genes. Now, the molecule I study, the ribosome, is what does that translating. It takes that information and actually makes the protein. But it doesn't do it directly on DNA. DNA is like, you can think of it as a library or an archive, and then you make copies of different parts of the DNA as you need them. And those copies of genes are called mRNA. Now, 3 years ago, nobody had heard of mRNA. But now, of course, after COVID, everybody knows about mRNA because of the vaccine based on mRNA, right?
Right.
But what that mRNA is simply the gene for it, a copy of the gene in the coronavirus that codes for the spike protein. And the way that vaccine works is the mRNA gets translated by my molecule, the ribosome, to make the spike protein and displays it. And then our immune system recognizes it as a foreign protein, makes antibodies to it. Then when the real coronavirus invades us, it's already prepared, with antibodies and an immune reaction to it. So you get the gist of it. The ribosome is at the very heart. It's at the crossroads between our genes and the products that they specify. So it's very, very fundamental to biology. So you can think of me as a molecular biologist who works on the ribosome. And that's not how I started off, but that's another story.
Yeah, well, that's great. You give a nice segue into it. I want to ask you about how you started out, briefly. I mean, obviously, it's it's hard to detail one's life.
I was born in India, and except for a year and a half when I spent in Australia, I basically grew up in India until I was 19. And at 19, I graduated with a degree in physics And I came to America, which was, to me, the land of opportunity and, excitement, etc. I'd been, I had a whole childhood full of Hollywood movies. So so this was the place to go to. And so I ended up at the age of 19 going to Ohio University to go to graduate school in physics. And I like to say the only notable thing I did in my 5 years in graduate school in physics is that I met and married my wife, because the physics turned out to be not for me, so I had no trouble taking courses and, doing well on exams. But once I started doing actual research physics, I realized, I wasn't working on a problem that interested me. And I thought, maybe I should look around and see what's exciting, what's going on in the world of science. And I used to read Scientific American, and I still do actually occasionally. And I noticed that there were lots and lots of articles describing big breakthroughs in molecular biology. That's when the sequence, first sequences of DNA were coming out. And that resulted in a Nobel Prize to Fred Sanger. And people were first figuring out what biological molecules like proteins looked like. They were understanding the immune system, the basis of the nervous system. The whole huge, big problems were, being cracked open. And so I thought, this is a revolution. I want to join this.
Yeah.
But I didn't know any biology. So I finished my PhD in physics because I didn't want to be a quitter and leave halfway through. But then I decided, instead of going on to the next stage, which is like a postdoctoral research fellow, I thought I should go back to grad school. And I applied to a bunch of grad schools. Some of them wouldn't even take me because they said, you already have a PhD in physics, so we can't take you as a grad student. But University of California in San Diego said, sure, you can come as a grad student and we'll take you. So they gave me a fellowship. By this time I was married. I had a 6-year-old stepdaughter and a 6-week-old son. Okay. And we had to pack all our stuff into a Ryder truck and drive it from Ohio to San Diego. That was quite a trip. Anyway, so I started grad school in biology. And, after a couple of years, I realized I'd picked up enough biology. In fact, I even took undergrad courses, even though I had a PhD. Because initially I didn't understand a thing, and so I had to start at the beginning. And then after a couple of years, I decided I was ready to take that next step. And so I wrote to these 2 professors at Yale who were working on the ribosome. And I said, how about, I come and work for you as a postdoc, which is the next step after a PhD. And actually, one of them had offered me not offered me, but said he would be interested in hiring me straight out of my PhD. And I told him, no, I don't know any biology, so I can't take that step. So I told him, well, you were willing to consider me when I didn't know a thing, know anything about biology. And now I've learned some biology, so maybe you're still interested. And so they hired me, and that's how I got into studying the ribosome.
And so you stayed with the ribosome, which is interesting.
Yeah, which is interesting. Most problems don't last 40 years or 50 years. Okay, you solve it and you move on, or you're really dotting the I's and crossing the T's, and so most problems don't last that long. But the ribosome is so complex. It has 500,000 atoms. Nobody knew what it even looked like, let alone how it worked or moved. So it just took a very, very long time to make those breakthroughs.
Yeah, you were talking about it a little bit earlier, but maybe you could just kind of land here for a minute and really just explain to listeners the makeup of the ribosome, how it functions. You talk about in the book these kind of small and large subunits, which are really important, and obviously with tRNA and mRNA. So maybe talk about the ribosome and how it works.
Okay, so let's start with DNA. DNA contains the gene, and the gene is basically like a string of letters. So the DNA is made of a four-letter alphabet. They're called bases. We call them A, T, C, or G. And the order of the letters is what specifies the information. It's like a sentence. The order of letters in a sentence makes up the words and makes up the meaning of the sentence, right? And so it's basically like that. So now you take a section of DNA that contains the information to make a protein, and you copy that. That's your mRNA. Okay? Now, in organisms like us, the DNA is tucked away in the nucleus of the cell. But the protein is made outside the nucleus in the cytoplasm. So the mRNA has to be exported out. That's what happens. Then when it's exported out, ribosomes find the mRNA. And there's a complicated mechanism where they figure out where to start reading the code. Now, the ribosome, as I said, is an enormous complex. It has about 80 proteins of its own. But it's 2/3 RNA. So you have an mRNA, which is one kind of RNA. There's a different kind of RNA that makes up the ribosome itself, the core of the ribosome. That's called ribosomal RNA. And when it reads the code, it's not, the building blocks of proteins are called amino acids. There are about 20 of them. And the code on DNA or RNA is read 3 at a time because there are only 4 DNA and RNA only have 4 types of building blocks. So if you read them 1 at a time, you'd only be able to code for 4 types of amino acids. So you have to read them 3 at a time. That gives you 64 possibilities, which is sort of redundant. But the redundancy is used in interesting ways. But how's that 3-letter on the mRNA recognized to bring in the correct building block of the protein, which is the amino acid? So it turns out there's like a bridging molecule. And that's another RNA called transfer RNA. And what that RNA has, it has 3 bases that read the code on the mRNA. So if it matches, that tRNA is accepted. And at the other end, it brings in the amino acid. Now, what the ribosome does is it assembles these tRNAs inside it, 2 at a time, and links in the amino acid from the new tRNA to the growing protein chain on the other tRNA. And then the whole assembly has to move. So it's like a machine where the tRNAs come in, deliver the amino acid, and then move through the ribosome and get ejected. And all the while, the protein chain is getting longer and longer, adding 1 amino acid at a time. So you can imagine, nobody knows what a ticker tape is anymore because everything's electronic. But think of it like a long paper tape in which all the information is written out. And you've got this little machine that's reading this tape, bringing in the components it needs to build up the protein as it goes. It's very, very complicated. And to your listeners, I'd say, if you sort of Google my name and the ribosome on YouTube and a translation, which is a movie about translation, we put a movie out there which shows how the ribosome works. And it's really amazing. You've got these 2 subunits which kind of ratchet, so it's like a ratchet moving along the mRNA. And, that's why you need the 2 subunits, is so that they can move relative to each other. And one of the subunits is the part where the code is read, and the other subunit is where the protein chain is made by adding amino acids. So that's another distinction between these 2 subunits. So it's highly complicated, very, very old machine. It's one of the oldest molecules in life. We have it. All species of life have ribosomes. They can't live without a ribosome. And even viruses which don't have ribosomes use our ribosomes to make their protein.
Wow. So, this is obviously essential for, as you're saying, all of life. I'm assuming unicellular and multicellular organisms. And so when you started looking at this, so this is the interesting thing I want to kind of figure out here, because the book does this where it does a lot of science, but it also is, your story as well. When you started doing this work in the 70s, 80s, whenever you started looking at this.
Yeah, late 70s, early 80s.
What did we know about the ribosome when you started? And where are
Yeah.
we now? Of course, you're a obviously central figure in this story, but when you first started out, what did we know? What did we not know? And what has that evolution been like of knowing?
We knew quite a lot. So, for example, we knew what the composition of the ribosome was. We knew that it had 2 subunits. It was itself made of 2/3 of it was made of RNA, but the remaining 1/3 was split up into about 50 or 80 proteins, 50 in bacteria, 80 in us. Our ribosome is somewhat bigger than bacterial ribosome. And so we knew that. And we knew about mRNA. We knew about the tRNAs deliver the building blocks of the protein to the ribosome. So we knew quite a lot. And we also knew another important thing, that lots of antibiotics work by blocking the ribosome, by blocking bacterial ribosomes. So I think we knew quite a bit. What we didn't know was how the ribosome does it all. I mean, how does it actually work? And the problem was you couldn't visualize it in detail because it was too big by the technology of it was discovered in the late 50s. And, and then people started working on it right from then. But, in the 50s, 60s, and 70s, and even 80s, the technology to try and visualize in atomic detail what the ribosome looks like wasn't there. And the analogy I use in the book is, imagine, if you're a Martian hovering above the Earth, and you saw these little objects moving around. In Manhattan, they would move in straight lines, but here in Cambridge, England, they'd be, wiggling around these winding roads. And you'd think, oh, these are interesting. And they'd stop, and then little objects would come out of it. And then when the little objects went back in it, it would start again. You'd have some idea. Then you get closer and you say, wait a minute, this thing requires gasoline and it puts out, carbon dioxide and water and some pollution. You still wouldn't know what it looked like. How it worked, right? But then you start looking at it closely and you realize it's got a steering wheel and 4 wheels, and it's got an engine and a crankshaft and all this stuff, and pistons inside the engines, or if you're a modern car, it's some electric motor. So then you'd understand how a car worked, right? But if you didn't know what it looked like, you'd be stuck. You'd have no idea how it worked. And so it's the same with chemistry and biology. A lot of science has advanced by being able to visualize it. We didn't know that cells existed. If we hadn't seen cells, we wouldn't understand biology, because we wouldn't know anything about cells and nuclei. And it's the same with ribosomes. So the ribosome was sort of like a black box. We knew it was made up of these things, and we knew what it did, but we didn't know how it did it. And we didn't know how antibiotics would block it. We just knew that they blocked it. So I think that it's that real understanding in sort of chemical terms, how a ribosome works. That was the big puzzle. And people just, didn't have the technology in the 70s to figure that out. Took a while to get.
Yeah. You mentioned that process, and you talk about something that was a little bit unfamiliar for me, so maybe you can explain it here, was this use of X-ray crystallography, right? Which is essential for studying and knowing the ribosome. Just tell us what this is, how this works, and why it was important for understanding the ribosome.
Yeah, so until recently, the only way to get atomic structure of something like the ribosome was use a technique called crystallography. So let me suggest to your readers, how would you see something that's really small? I mean, I talk about the ribosome as very large, but it's tiny. In everyday terms, it's absolutely tiny. Small, much smaller than a cell. It's just large in molecular terms. So if we wanted to see something small, what we would do is we'd take a magnifying glass and magnify it. Or we would take a combination of lenses like a microscope and magnify it even more and see it in detail, right? But it turns out that there's a law in physics which says you can't see the kind of detail that's smaller, much smaller than the wavelength that you're using. Okay? And the wavelength of light is, it's measured in nanometers. A nanometer is 1/1000th of a meter. Okay, a meter is roughly a yard for American users. So anyway, it's about 1/1000th of a meter. Now, light is about 500 nanometers, okay, in wavelength. Now, the distance between atoms is like 1/10th of a nanometer, okay? So, light has way too big a wavelength to visualize the kind of detail where you would see atomic detail. So the way that to get around it is to use a kind of radiation that has a much smaller wavelength, and that's X-rays. Now, X-rays are basically light. They're the same physical object as light, except their wavelength is much shorter. And that wavelength, so they're much higher energy than light. And that's why X-rays can damage us. And so, we have to protect ourselves against X-rays. But otherwise, they're basically light. To a physicist, there's no distinction. They're photons. But the trouble is that there's no good lens to magnify X-ray, to magnify an image with X-rays. And even if there were, the signal from a single molecule wouldn't be enough. By the time you got enough signal, you'd have fried the molecule, because X-rays, as I said, are damaging. They would just basically destroy the molecule. So the way around that was let me back up and say, how does a lens work? Well, what a lens does is it takes scattered rays from the object and recombine them into an image. For example, when you see the image on the screen, and when you're looking at me, the scattered rays from the screen go into your eye. Your eye has a lens, and it combines them to form an image on your retina. Okay?
Yeah.
Now, let's suppose you weren't there. The scattered rays would still be there. They'd have the same information. Okay, so the lens is not creating the information. It's simply using the information. Okay? So what people figured out was that if you crystallize a molecule, and what crystallization means is you produce a very ordered stack of the molecule. Okay? And this is a bit like, every child knows how to crystallize salt or sugar. You just take a solution and let it dry out, and you get crystals. Okay? But if you have a big, complicated molecule like the ribosome, it's hard to get crystals, and people use various tricks to try and get it. But basically, you're coaxing the molecules to come together into a very regular stack. And it's like a three-dimensional stack, right? And then you shoot that with X-rays. What happens is, because you have this very ordered stack, you get a phenomenon where rays add up along certain directions, the X-rays. So instead of scattering all over the place, they're scattered along certain spots. And you can measure those spots, okay? Now, when you measure those scattered rays, you have the information that a lens has. But you could do what a lens does, which is recombine them. And what you can do that mathematically. So what a lens effectively does is a mathematical operation of recombining the rays. And knowing what the lens does, we can do that in a computer. So we can do in a computer exactly what a lens does. And so effectively, you're sort of using X-rays and using crystals to generate the scattered rays, which you can then use in a computer to combine it to form a three-dimensional image of the object. And that's how crystallography works. It's a very sort of, non-technical view of crystallography. Crystallography is highly mathematical, but I've just given you a kind of concept of how basically how it works. And so that was really, the method that was used to solve the ribosome. And why did it take so long? Well, a couple of reasons. One is, first of all, it's hard to get crystals of the ribosome. And one of the people who started that field was Ada Yonat, with whom I shared the Nobel Prize, and about whom I talk a lot in this book. And of course, the book, I have to say, is very, very frank, about people, so I don't pull any punches as far as, what I think of various players. But there's no question that Ada Yonath was the first person to get real crystals of the ribosome, and that's what got the ball rolling. But the other thing you need is you need these crystals don't diffract, don't produce a strong enough signal. And so to get enough signal, you need extremely powerful sources of X-ray. Now, a lab X-ray machine is quite a bit more powerful than the X-ray machine in your dentist's office, okay? but it's still not that powerful. So what we have to do is we have to wait for the development of synchrotrons, which are these very, very large particle accelerators. They accelerate electrons round and round a ring at the speed of very close to the speed of light. And in doing so, they emit extremely powerful fans of X-rays. And you can take some of those X-rays into a beam and focus them into a very concentrated beam and hit your crystal with that. And that produces enough signal to get the kind of data you need. You also needed very sophisticated detectors to detect the X-rays. And one interesting thing is that the people who got the physics Nobel Prize the same year we got the chemistry one had actually developed these CCD detectors. And of course, they're used not only for X-ray detectors, but they're also used in all our cameras and everything, so it's really kind of funny. We wouldn't have been there if they hadn't been, done their job. Nice. So we had to thank them. Anyway, so that was the other thing. And then another thing that was really important is the development of more powerful computers and computer graphics. Because the kind of data involved in collecting ribosome, they're like there are millions of data points to try and get 500,000 atoms in position. And so you needed, much faster computers and faster graphics. And all of that sort of developed while we were doing it. So we were using cutting edge technology as we were going.
With this, so this is what's interesting. I'm glad that you brought it up. You mentioned in the book that you can talk about some of the specifics here about ribosomes crystallizing. You mentioned specifically the crystals of GH5 and S5. So if you want to be specific, you can. But you mentioned how important it was to go to the LMB in Cambridge, right? And how there is where you start to kind of not build, but kind of show the building of the ribosome structure. So tell about that experience.
Yeah. So I did my postdoc at Yale, and I was using a very esoteric technique called neutron scattering. And that was a technique that physicists have used a lot. But in biology, its use has been quite limited. It's not really taken off as a big technique like X-ray crystallography or electron microscopy or things like that. But anyway, so after my postdoc, I had a lot of trouble finding a job. In fact, I applied to like 50 universities. I didn't get a single interview. Okay.
Wow.
And then I got a job for a brief time at Oak Ridge National Lab, but that didn't really work out. And so I then moved to Brookhaven National Lab, where, that's where I really started my career. But after a few years of using these techniques, I thought, look, if I really want to understand how the ribosome works, I need to understand how to do X-ray crystallography. And I don't know how to do it. So I convinced my department that I should go on sabbatical. And I wrote to Aaron Klug, who was the director of the Medical Research Council's Lab of Molecular Biology in Cambridge, England. And the reason I wrote it is that's the birthplace of crystallography. First of any crystallography was, in 1912, when Lawrence Bragg figured out how he could do structures by crystals, by using crystallography. But then to use it for proteins, a guy named Max Perutz, who started the Laboratory of Molecular Biology, which is the LMB, he's the guy who first figured out how to solve protein structures using crystallography. So I thought this would be a great place to learn crystallography. Of course, my wife and I, even though we're Americans, we're kind of Anglophiles. We like Monty Python and, watching Masterpiece Theater and stuff like that. So I thought it'd be cool to spend a year in Cambridge,
Yeah.
So I went there, and the lab was it was kind of weird. If you went from a well-funded American university to the LMB as it existed. Now we have a fancy new building, but in 1991, it was a shabby old building. There were like equipment in all the hallways because it was overcrowded. Probably in America, it would have violated all sorts of fire codes. and and everything looked a bit shabby. But the weird thing was, almost any equipment you needed, they had. It was all tucked away somewhere, squirreled away in a basement corner or something, but it was there.
Mm-hmm.
The other interesting thing I noticed there was people didn't waste their time doing like what I call, routine science, okay? They're all, working on big problems. And part of the reason was that the place had very stable funding. And so, they were not afraid to, work on the really tough, challenging problems. And also senior scientists would often work themselves. They wouldn't just sit in an office and, direct other people. So it had a lot of, it had a very special culture. And my host was a fellow of the Royal Society. He was like, well-known scientist. And when I arrived, there was no desk for me. So I said, well, how about if I just, use a desk in your office just for, a little while. And he kind of smiled at me because he didn't have an office. Here was this famous guy. He had no office. He had a little desk himself. So it was a very strange place. And so I had a very good time. And I took two small, data from two small protein crystals to learn how to solve structures using crystallography. And I'd already collected the data at Brookhaven, so I didn't have to actually do any experiment, but I'd learned how to process the data, how to analyze it, how to build a protein structure, et cetera. And that's what gave me the idea that actually you could use a particular method in crystallography. There are different approaches, and you could use a particular approach even for a very big thing like the ribosome. So when I finished my year, I already had this germ of this idea that actually it ought to be possible even to solve something really big like the ribosome using this method.
That's so interesting how, we have these ideas about these, various places, and then, it's always not quite the same, but it sounds like the quality was there. And that's nothing to say about American universities or anything like that. But
no. I mean, America has fantastic science. But I think the LMB, which is the Lab of Molecular Biology, the LMB has a special history, and also it's trained, I would say, many American Nobel laureates were postdocs at the LMB. It has this amazing influence, worldwide influence on molecular biology. I mean, starting with Watson and Crick and Sanger, figuring out how to sequence DNA and the first virus, organizations, lots of things, antibodies, and most recently electron microscopy, which has taken the world by storm.
Wow. So
it's had a long history of really one big breakthrough after another. And I should tell you another thing that I learned there. So I would go to seminar. This would be when visiting scientists would come and talk about their work. And there are all these famous Nobel laureates sitting there in the audience. I was in complete awe of them. And then they would ask the dumbest questions after the talk, like things that seemed really simple to me. And I realized that just because, they're smart at one thing, it doesn't mean they knew everything. And they didn't care about showing their ignorance. They realized that, being ignorant is nothing to be ashamed of. That's why you ask a question, So it made me realize, that just because you may be an expert in your field doesn't mean you know everything. And also you should have some humility and not be worried about asking questions, if you don't know. And that was another very interesting lesson. These people were not, they were not kind of stuffy.
Mm.
You know,
Well, that has to be, again, such a good model for young scientists and for people that are trying to figure things out. This kind of way of having this kind of epistemic humility of like, yeah, I don't know everything, and it's fine to say I don't know everything. I know
exactly.
a few things in my field for sure,
Yeah.
but I'm not going to know everything. I think
Yeah,
that's
and
what
they
makes you a
weren't
good
afraid of
scientist.
asking the simplest things. Even the simplest question, they were not ashamed to ask it. And so that really was an eye opener, and I thought that was great.
Yeah. So I want to you talk about in the book. I mean, what was this experience like for you to see an RNA helix of part of a ribosome for the first time and seeing the atomic structures of those subunits? Tell me about that.
It's totally amazing. So what happens is, all this hard work, making the crystals, getting special atoms that you need, which have some properties for the X-rays, which allows you to solve the structure. Taking it all, I mean, the crucial data set that we collected, we had to go to Chicago to collect it. So we took our crystals in these liquid nitrogen containers, on the airplane. Luckily, this was, well, it was 2000, so it was before 9/11. And so otherwise, who knows what the situation would be now. Anyway, we took them all, on the airplane, checked them all in, and then drove from Chicago Airport to Argonne National Lab, which is about 20 miles away, and which had what was the most powerful synchrotron in the world in those days, and also had a very special instrument that allowed us to align our crystals very precisely, and we needed that for a technical reason. And anyway, so we got there, and the way it works is you're given 2 days of X-ray time, instrument time. So, and the X-rays are produced round the clock, so you can't go to sleep. You have to work in shifts. So, we worked in 12-hour shifts. We took 4 people, and 2 people worked the day, 2 people worked the night, and actually we kind of staggered it, so there's always some overlap. And after 48 hours, you don't know if your experiments actually worked until you do the calculation. And once we did the calculation, and then actually did the calculation to calculate the map, it's really an amazing feeling, because then you say, wow, you can see all this detail. And then you still don't know what the structure is. You have to build it into this image. And it's a bit like solving a jigsaw puzzle. You know what the pieces look like, and you have to figure out how the pieces go in this image, and then you end up with a structure of a molecule. But just seeing that image and seeing, RNA helices and proteins attached to them and all these, sneaky things, it was really pretty amazing. And I like, in the book I describe, it's like seeing a new continent. You're seeing something that nobody's seen before.
That's so cool.
Okay? Nobody in the world has seen this particular thing, which is at the heart of biology, right? So it's a big rush.
Well, and also knowing how consequential it is. It's not like it's just seeing like, a different side of this coast of the coastline of a country that we already know exists. Like you're seeing something the first time that's super essential for all living things on the planet. I mean, that's incredible.
Yeah. In fact, I say this in my book. I'm not exactly a jump up and down kind of guy. I'm usually, especially in the lab, I'm pretty calm. Most of the time. Although my family probably would disagree with that. But anyway, when we did this calculation, we'd made a mistake, and when it spat out the results, we thought the experiment had failed. And then I noticed a little error in our script, that we had submitted for the calculation, and we corrected the error. And then as soon as it spat out these numbers, we knew the experiment had worked. And, I had this very reserved English technician. And when I saw it at work, I couldn't contain myself. It was like 48 hours of, sleepless stuff and months of, tension going into that thing. Suddenly, like, wow, it's work. And I just got up and I started dancing around the room saying, we're going to be famous. We're going to be famous. The guy thought I was nuts, because I think, he realized the ribosome was important, but I'm not sure he realized quite how important it was, So
it's amazing.
Anyway, it was pretty amusing.
That's so amazing. And on the cover of the book, this is the ribosome, right? This is a picture of the ribosome.
It is a picture of the ribosome that I generated that picture using computer graphics. So.
Wow, it's so amazing how it looks at this level. Of course, it's super small, but when you blow it up,
and that I should say is a highly simplified representation.
Really, wow.
Because it's actually made up of 500,000 atoms. If I were to show you all the atoms, it would just look like a mess of atoms. You wouldn't be able to make any sense of it. So so people try to simplify their representation of protein, so you can sort of see something about the architecture and how it put together.
Well, it's interesting. Just kind of on the face of it, it just seems very. It doesn't seem like there's a lot of contours to it. It's very sprawling. It's very out there. It's a lot of. It's got its like tentacles and everything, which is
Unlike DNA, DNA is a very simple and beautiful molecule. The ribosome is like something that sort of grew into this kind of monstrous thing,
yeah. So you knew you were going to be famous. You knew you'd be in the
in a limited way.
Right. You'll be in biology and chemistry, history forever. But how is that like process then of like, the afters, right? Okay, you see this, you have the team of folks that see this, and then getting it published and getting people to talk about it, and then finally this whole idea of people you talk about in the book. I didn't know all this it's like a process of the Nobel Prize. It doesn't just like happen overnight. It's like a whole thing.
No. So first of all, the structure was only a beginning, because once we had the basic structure, we could figure out how antibiotics bind to it. And that's really very straightforward. So these antibiotics were discovered in the 40s and 50s. And, now for the first time, we could actually see them in action. See how exactly how they block the bacterial ribosome.
That's amazing.
And why it's safe for us to take them, because they don't bind so well to our own ribosomes.
Wow.
So they don't poison us, but they poison bacteria by stopping bacteria in their tracks. So that was another excitement. Then figuring out how the code is read and why is the code read so accurately. That was another big step. And then over the years, we then started working on, first I worked on the small subunit, but then I started working on the whole ribosome and figuring out how it moves and how it terminates and how it starts. So all these things, different steps, took years and years. And so we were doing science, one thing after another. It wasn't like we were sitting around after the first structure waiting for recognition.
You put your feet up and you just said, "Don't wait for all the phone calls." You had to keep working, right?
It wasn't like that. No. We were working, working our tails off. So, so I think, and it's not just because, it's because it was exciting to find out the next step and the next step and the next step, because figuring out how this machine works, okay? And it's like getting snapshots of the machine in action so that you can kind of make the movie of the machine working, So I think that's what we were after. And that each one of those projects took, sometimes several years, So that was what we were doing. But in the meantime, people were already getting excited that this was this big discovery. We were getting invited to all kinds of meetings. And from 2000 onwards, I was getting invited to meetings in Sweden almost every year from 2001 to 2004. Okay.
Wow.
And in 2004, there was this huge meeting. Well, it was not huge in numbers, but it was like a very important meeting. Lots of famous scientists, people who discovered how DNA replicates, and how DNA is copied to RNA, and the ribosome, all sorts of related fields. There were all these famous scientists there, and you could tell that this was Swedes basically taking a look at us,
Mm.
and it was like an audition, I think,
Mm.
and so anyway, and often there'd be members of the committee there, and sometimes I didn't even know who these people were, and and then other people would say, "Hey, did you know those people are on the committee?" Things like that. And you could also see how scientists, famous scientists,
Mm.
would act like really nervous, like graduate students before a qualifying exam. They were like really nervous giving their talks, and because they sort of knew that there was quite a lot at stake, because they were being sort of judged, and it was a very weird feeling. These people were all big shots, Many of them they wouldn't give you the time of day normally, and here they were really nervous because they were in Sweden before people in the committee. So that was sort of an interesting sociology. And so, often the same gang of people would go around the talk circuit. There'd be like different conferences, different parts of the world, and see the same people giving the same talk. It was a bit like almost like a campaign in a way. And in 2004 was my last one. And then I decided, for reasons I go into in the book, I decided that, the hell with this. I'm not doing this, anymore. And I basically stopped going to Sweden for almost 5 years. Except I only gave one talk, and that was because a friend of mine invited me to his institute. But other than that, I just didn't go to any meetings there. And so I was kind of surprised, in the end, to be one of the three, because a Nobel Prize has this other limitation that they only give it to 3 people at most. So if more than 3 people have competed, not competed, have contributed to the process, then it becomes who's going to be the 3 that get that phone call. So it's a bit like musical chairs. Who's going to have the 3 be sitting in one of the 3 chairs when the music stops? So I didn't think I'd be one of them for complicated reasons. But so it was a bit of a surprise.
And so what was it like to get that phone call? Where it's 2008 or 9 or whatever it was, and
in 2009, they only give you about half an hour's notice. So they call you half an hour before they go
live with the
with the announcement.
Oh, wow!
And they in fact, the guy said, "Enjoy your last 30 minutes of peace."
Everybody around the world's calling you and emailing you, right? Yeah.
So, but it was quite funny because I didn't believe it, and one of the reasons I didn't believe it is in that meeting in 2004, there's a very famous ribosome biochemist in Sweden, and he was at that meeting, and he had a big disagreement with me about a key aspect, how the ribosome is accurate in reading the code. And so he came up to me after the banquet and had a big fight with me, And then a few months later, I found out he was on the Nobel Committee for Chemistry. So I thought, well, there's no way this guy is going to pick me as one of his 3 because, he doesn't. He really dislikes my work, So I sort of gave up on it. And so when I got the phone call, I didn't quite believe it. I thought maybe it was a prank. I have some friends who might, convince some Swedish guy to call me in and pretend, it was a Nobel committee. So anyway, so I told the guy, look, why don't you put this guy on the phone? Because if it's real, then he's a member of the committee, so he's probably there.
Right.
And then this guy, Mons Ehrenberg, came on the line, and that's when I knew it was real.
Oh, wow.
And and you have to hand it to the guy because he put aside his differences about one small aspect of the work and looked at the big picture. And he realized that in the big picture, he felt in the big picture I had made a major contribution. So he put aside his little disagreement. And many people wouldn't have done that. They would have been petty about it. They would have let this personal feeling get in the way of being objective. And it shows that these people really have integrity. I mean, they're humans, so they can make mistakes, and people can argue about their decisions. But I don't think they're dishonest, and I don't think they're political. And that's one thing I do believe about the Nobel Prize.
You can tell me about, like, a little bit about getting the award and the ceremony and the whole thing. But I mean, when did it kind of hit you that you won a Nobel Prize? I mean, when did it, like, I mean, there's the phone call, but then there's like all the afters, and then everything's kind of died down. But when did it hit you?
It hit me, I guess, when I saw it on live online and saw them actually announcing it.
Wow. Yeah.
And then this deluge of phone calls. They had to basically cut my phone line off, and for 2 days. It was nonstop for 2 days.
Wow.
And even my wife couldn't get through.
Wow. What was it? I guess, I mean, you're from India. I mean, what was that like, to be from India to get a Nobel Prize?
It's kind of funny because, I had become an American citizen in the mid-'80s.
Sure.
And I had not really gone to India very much at all. Or in 30 years, when I lived in America, I'd only been to India 3 times.
Wow.
And I'd left India when I was 19. So I didn't have a lot of connections with Indian science until the early 2000s. When I was in England, I went to my first meeting in India, a scientific meeting. And then that made me connect with a bunch of Indian scientists. And then I would visit every year or every other year. But by and large, except for the science community, the Indians basically ignored me. So when I became a national member of the U.S. National Academy of Sciences, there was nothing in India saying, about me. When I became a fellow of the Royal Society, there was nothing, which is the British equivalent. Actually predates the National Academy. So, Indians by and large had ignored me, except for the people in my field who knew that what I'd done was important. And so as soon as the Nobel Prize hit, suddenly everybody in India was, sort of felt like, oh, this is our boy, he's won the Nobel Prize. And I felt a little odd, because I hadn't lived there for 40 years and nobody had paid any attention to me. And suddenly I was getting letters from government officials and the prime minister and the president and all these people congratulating me. And then I realized, well, actually, it's okay because I grew up in India. I was educated in India. I did my whole undergraduate education in India. The Indian government paid for, I had a free education. They paid for my whole tuition and they paid me a stipend. And then I left. So I do owe India quite a lot for launching me as a, educated to the point where I could go to grad school. So I thought, that's okay, and I'm as much a product of India as I am of America or of Britain. Each of those 3 countries has really contributed something really significant to my life and career.
Yeah, I mean, Nobel Prize, but I mean, then there's all these. I don't know how you feel about this. I'm curious. You get all of these things. If people look you up online, you have so many awards and honors. I mean, one of them is the Order of Merit, which is like been given to like 20 people or something, right? Like it's crazy.
Well, yeah, the only 24 people. It can only 24 people at any one time can have it.
Right,
Somebody has to die.
That's what it is. Yeah, that's what it is. Yeah. Which is, I mean, that's wild. I mean, some crazy people on that list and all these other things. And it's just, I don't know. What does it Does it feel kind of surreal in some ways of like?
It is pretty surreal. And I have to say, luckily my family doesn't treat me any
That's
differently.
good. That's good. They keep you honest.
Still
They
have to
keep you
do the
honest.
dishes. So, I don't get any slack.
That's good. That's good. They keep you honest. That's good.
They keep me honest. And my lab keeps me honest. And the L&B is one of those places. So at many universities, if you win a Nobel Prize, you suddenly become a big deal. You get a free, like at Berkeley, I'm told you get a free parking space.
Right. I've heard this. Yeah.
And other places, you have a street named after you.
Right.
And in the University of Utah, there's a street named after Mario Capecchi, who won the Nobel Prize. Here at the L&B, they've won so many Nobel Prizes, they don't really care. And I don't even get a bicycle parking space. If I'm late for work, I have to hunt around for a place to lock my bike. And a few days after the Nobel, my director comes up to me and said, I know you've been a little distracted, but your report is overdue. so there's no particular thing at the lab either. So,
no,
that's I think healthy, And by the way, science is a healthy environment. So just because I have a Nobel Prize doesn't mean it's easier for me to publish.
That's good.
In fact,
that's good though.
I've noticed it's actually almost harder to publish because the anonymous reviewers say, "Hey, this guy thinks he's some big shot. We're going to hold him to high standards." I think there's some element of that because it's been just as difficult to get our papers published. Yeah. So I think science, basically, science has all sorts of problems. Fundamentally, it's a field that has integrity and keeps people honest. It doesn't mean everything in science is honest.
Sure.
There's obviously all kinds of problems and fraud and so on. But the process and the system really is based on the fact that people have integrity.
Yeah. What is it that you're working on now with the ribosome, and where do you want to see, or where do you hope to see or predict to see the future of work on the ribosome?
Yeah, so I should say I'm only a couple of years away from retiring.
Sure.
And one of the things that we're trying to do is, how do ribosomes know when to start and where to start on the message?
This agency,
so it's not at the beginning.
This agency kind of factor of sorts, right? Of like, what's pushing it to know now versus not, or things like that, yeah?
Yeah, so that process called initiation is highly regulated. So you can make initiation efficient, you can make it less efficient, you can disrupt it, and viruses will disrupt our initiation. For example, the first thing coronavirus does is it stops our ribosomes from initiating on our own mRNA. And it has a trick that allows ribosomes to make the viral mRNA, translate the viral mRNAs, but not translate our mRNA. So that's just to give you an example. So that whole process of initiation, how it works, how it's regulated, that's a big complicated problem, and that's one of the things we're working on. One person in my lab is very interested in a problem called quality control and stress. So when cells starve, for example, if they're not enough nutrient, let's say they don't have amino acids to make protein, why would they, continue to translate? So they stop things. They stop the process, and then they divert it to stress response genes. And how that works involves the ribosome. If the ribosome is stalled because it can't get tRNAs with the correct amino acid because there's a shortage of amino acids, or there's other kinds of stress, or the mRNA is defective. There's all kinds of problems that you can encounter in the process. And the cell has elaborate quality control mechanisms, many of which involve the ribosome. And so that's another area we're working. And another area that's of great interest for therapy is circular RNA. So normal RNA is what's called a linear RNA. It's like a string with two ends. Now, if you take those two ends and join them together and make a circle, that would be a circular RNA, but that's that's not normal. That's not the way most translations work.
Hmm.
But the reason circular RNA is interesting is because it's very stable, because most RNA gets broken down from the ends.
Hmm.
There are little protein enzymes that chew up the RNA from the ends. And if you have a circular RNA which has no ends, it'll be very stable. So we're trying to understand how translation works from these circular RNAs. Because that could be very important for mRNA-based therapy,
Hmm.
where you're supplying mRNA, which codes for a protein that could be defective in someone. Somebody has a genetic disease, and you could supply the mRNA with the correct form of the gene and make the functional protein that would cure their disease. So it has important implications in therapy. So that's another area where we
I was gonna ask about that, is, where we have things like various viruses such as coronavirus, things like cancer, or obviously ribosomes are implicated in many things. It does seem like the more we learn, there's more windows that open for trying to understand how we combat certain things or cure things or of this nature. Yeah.
Oh, absolutely. Absolutely. And understanding how ribosomes translate mRNA is key to really making mRNA therapeutics really effective. It's worked for the vaccine, but vaccines are a very limited use of that potential. The potential is far greater. It can be used for correcting genetic diseases. It can be used to target cancer cells, as cancer vaccines. So there are many, many uses of mRNA therapy. And understanding like circular RNA translation could make that therapy even better. And so there are lots of things to study. And of course, designing better antibiotics.
Sure.
That's another big thing. We're worried about. We saw what COVID did to humanity during the pandemic.
Yeah.
But if we get a bacterial plague, that could be really, really serious.
Yeah.
And if it's resistant to antibiotics, we would have no recourse. So I think, we need to try and stay ahead of the game by having good antibiotics in our arsenal in case we have these, bacterial pandemics. So, there's lots of things. And of course, it's also fundamental biology. It's about how the cell regulates protein synthesis. It's involved in lots of things like aging and memory and Alzheimer's and all sorts of things. They all involve protein production. And if we can somehow control or understand how that's regulated, we can start tackling many of these problems.
Yeah. Well, my final question for you is you've made so much contribution to science and for many generations after you. When you look back on your life and both professionally and personally, what are the things that you consider? I mean, you come from India, you've lived in America, you lived in Britain. I think it's very interesting that you you have a PhD in theoretical physics and you started in physics, but your whole life has been in something else. And I have to imagine that perspective is you probably helpful in some way of just your straight biology. And I'm sure there are many other things. How do you see your life in these ways?
I think, if you ask, why did my life work out? So before I go to that, I want to say a little bit about the book that we haven't talked about, which is that the book is a very frank look at how science and scientists work. It's not just about the ribosome. The ribosome, they're kind of two parallel stories. There's the ribosome story, but there's also what scientists are like. We're competitive, we're altruistic, we're jealous, we have egos, but we're also friendly and nice to each other. This complicated mix of humanity that makes up scientists, that is part of the story as well. And so you ask about my own story, and one thing I would say is the reason that I might have succeeded is because I've been open to things. I've been open to changing fields if things didn't work out. I've been open to changing institutions if I wasn't quite getting what I wanted in one place. I was not afraid to move. And wouldn't even mind moving to a different country.
Mm.
I took a huge salary cut to come to England. And, you have to sort of ask, what do you want out of life? And somehow make that possible.
Mm.
And to do that, you might have to be open to change, different circumstances, etc. The other thing is science doesn't pay a lot, but it's a lot of fun.
Mm.
Every day you come, you're trying to do something new. It's also international. You meet people from all over the world. My lab now has somebody from Spain, Germany, China, America, India, and Ukraine.
Mm.
Okay, so that's sort of my lab. Okay, so I think it's this highly international endeavor, and it makes you realize that humans may have cultural differences, but underneath, we're all the same, and we have the same aspirations. We all want to know what's happening. We want, to look for the truth in science, and we all have the same aspirations. Fundamentally, we want to, do good work, have families, etc. So I think it gives you a really broad perspective on life.
Yeah, no, that's very nicely said. Well, the book is called Gene Machine, The Race to Decipher the Secrets of the Ribosome. This is out through Basic Books. It's out in paperback now. Obviously, you got a lot of things going on. Is there any place you want to point people to in particular, your lab, your research, any place you want folks to get at?
I think if they Googled my name, Venki Ramakrishnan, it would direct them to our lab website. And then they can sort of see what's going on. The one thing you won't find in the website is my new book, which is a book on why we age and die.
Nice.
It's called Why We Die, and that's coming out in March.
Oh, very nice. Very nice. Well, Venki, this was such a wonderful privilege and honor. I really do thank you.
It's a real pleasure.
Yeah, thank you.
No, it's my pleasure.
So much
Thank
for
you.
coming on. I greatly appreciate it.
Thank you.